Introduction. For the next and final section of the day, Animal Health, are we winning the battles but losing the war? And nos prochains panélistes vous présenteront des...
Our next panelists will present the results of their animal health projects, projects that are once again part of the research cluster. I'm delighted to call upon the first panelist, Dr. David Kelton,
at the Dairy Farmers of Ontario Dairy Cattle Health Research Chair in the Department of Population Medicine, University of Health. His research interests include infectious and metabolic diseases of dairy cattle, including particular interest in Johne's disease, bovine mastitis, and bovine lameness. Welcome.
Thank you very much, Shelley, and it's certainly a pleasure and a privilege this afternoon. I thought it was interesting when I was invited to participate in this panel that the section heading for this is Are We Winning? And it's apropos that we lead off this session with a discussion of the National Dairy Study that hopefully most in the room have at least heard of, because I think this may give us an opportunity to figure out whether we're winning or not. Because my first thought was, how do we know? We need to be looking at where we are and understanding where we are as a dairy industry in order to decide whether we're winning or not. So bear with me. What I would like to do is I'm going to start with a question. There we go. One of the elements of proaction is biosecurity. And the reason that biosecurity is of interest, I think, is that as an industry, animals move between farms and within farms a fair bit, and we worry about disease transmission. It's one of the elements that we looked at in the National Dairy Study, and the first question I have for you is, I'd like you to tell me what proportion of farms across the country, based on our estimate, had introduced at least one live animal onto the farm during the calendar year 2014. And so your options are less than 10% of the farms in Canada, 10 to 25, 25 to 50, or greater than 50%. Wow, this is fun. Okay, looks like it's stabilized there, and we're sitting 25 to 50 or greater than 50%. The answer is actually just under 50%, or about 47%. So pretty much pretty close to the target there. I'll come back and talk about that in a little bit of detail. But I wanted to start by giving you a little bit of background of where these data came from and how we gathered them and why we gathered them. How many of you in the room are familiar at all with the National Animal Health Monitoring System in the United States? Show of hands, just quickly. A few of you. So it's a system run by the United States Department of Agriculture. They run a national benchmarking study every year. They've been doing so for about 35 years, and each year they focus on one particular commodity group. And so they've done, in 2014, they did their 6th dairy study, and they come about 5 or 6 years apart. The unique thing about that is that they are national in scope, and they've allowed them to benchmark or determine what's going on in the dairy industry across the U.S. and repeat that on about a 5-year cycle to actually not only measure what's going on, but whether things have changed from one survey to the next. This is something that up until now we really haven't done in Canada. We've done a lot of great work. A lot of it has been done regionally or has focused on particular issues. We had the production limiting diseases surveys that were done 10 to 15 years ago, targeting particular diseases like BVD, Johne's, and Neospora. The Bovine Mastitis Research Network that Simon will talk about has certainly looked nationally at mastitis, but again in that narrow context, and we've had some other opportunities. But even in some of those issues, like the ONI's programs that have varied geographically, we've used some different methodologies. And so trying to bring that all together and paint a national picture has been a little bit of a challenge. So about 5 years ago, I was having a beer with Jason Lombard. Jason is the veterinarian who works for the USDA in the US and actually runs the NOMS dairy studies. And we said, we decided, wow, wouldn't it be fun to do a Canadian study about the same time they're doing their next US NOMS study, so we could compare, not only look at what's going on in Canada, but do some cross-border comparisons. That led to the development of the idea for this project and actually brought that to fruition. And I'll take this opportunity to acknowledge the help that we got from Jason Lombard and his team in Fort Collins in just giving us some ideas about methodology and so on. In trying to do a national study, we had 5 broad objectives in mind. First was a bit of a benchmarking exercise, so to collect some baseline data on health productivity and management. The second was to look at some key diseases of economic or zoonotic importance. The third was to look at some of the relationships and some of the regional differences, perhaps in management and disease prevalence. The fourth was to document the uptake of some key best practices in the areas of milk harvest, biosecurity, and animal care. And then finally, to produce some results that were comparable. And again, we used a few of the same data collection mechanisms that they did in the NOM study in 2014 to be able to do a little bit of cross-border comparison. Our industries are different in some respects, but similar in others, and we want an opportunity to look at how they might be the same and how they might be different. The study was really laid out in 3 elements, and I'm not going to spend a lot of time on this particular slide and go into a lot of detail. We started in 2014 with a needs assessment, and when we had over 1,000 stakeholders in the dairy industry, hopefully many of you in this audience participated in that, where we tried to get a sense of what the major areas of interest and focus should be for this kind of a national study. We had a great response from dairy producers and other people, other stakeholders working in the dairy industry. That then set the framework by which we developed the national study that took place in 2015 through a broad-based questionnaire and then some farm visits that happened last summer. And then we're into phase 3 now, where through the fall we've been pulling all of the data together, cleaning it, validating it, and now literally in January of 2016 we're starting on the analysis. And I'm going to present just a very small number of some of the highlights for you, because we've been asked to present some of this to the ProAction working groups as they move ProAction forward. On the needs assessment side, it was very interesting. We divided our responses into producers, people that worked for government, either provincial or federal, and in the bottom is the other category, a lot of people working in nutrition. That included veterinarians and others. The key thing here was the number one priority that was common to all 3 groups was the issue of animal care and animal welfare. Probably not surprising, because at that time there's a lot of discussion about the Dairy Code of Practice and the development of the animal care assessment through ProAction. As far as the major diseases of interest, again, there's quite a collection across there, and we've tried to look at a number of those in the dairy study, but also interesting that the top priority across all of our stakeholder groups was lameness, and again, nice tie-in there with animal care and animal welfare. So we took all that input from our stakeholders and we developed a questionnaire. It was a bit of a monster of a thing. 113 questions, 49 pages, and really just scratched the surface. We had a lot of breadth, not necessarily a lot of depth. We were looking for input on some of the key elements that had been identified in the needs assessment, and also to partner with some of the other projects that were going on in this dairy research cluster too. So we got some questions from one of the grad students working with Ronaldo Seri, also with a number of the other groups, Filippo Miglior's group on lameness and so on, to try to give them a broader national perspective on some of the things that they were looking at in a little bit more detail. The questionnaire was available in English and in French, primarily as an online tool, but also if at the request of the producer they could get a paper copy or in fact complete it by phone interview. Given the scope of this study, I'll take this opportunity as well, because I know they're representatives of milk producers and dairy boards from across the country. We had a fantastic cooperation with all 10 provincial milk boards. To make something like this happen, we actually had to access producer lists and de-identify the data so that we didn't have any individual identifiers for those dairy producers, and the only way we could do that was through the milk board, so we appreciate that. And our access to those was based on our regional relationships, and there's one picture missing on the west coast there. There's supposed to be a nice picture of Herman Barkema, but you can see him in person. Oh, it's on that slide, but not on this one, okay. Jocelyn Dubuc in Quebec, and Greg Keefe out in Atlanta, Canada. So again, a big thank you to the provincial milk boards and the producer associations for their cooperation in all of this. The Phase 1 questionnaire, originally we had talked about selecting a random sample, and at the end we decided, well, let's invite all, at that time, 11,962 licensed producers in the country to participate and see what happens. The day we hit send on the invitation, I'll tell you, was a sleepless night. I woke up and my recurrent nightmare was that nobody would answer, right? This long questionnaire, what kind of a response we'd get. We got over 1,300 dairy producers across the country in all 10 provinces to participate, a little bit over 11%, which is phenomenal. I mean, in my best hopes, I think we targeted about 1,000. So to get the response we did was fantastic, and we're very grateful for that. As I said, we're pulling this together and just getting to the point now we're starting to look at the data and what we're learning from that. But an interesting thing was when we look at the demographics of the producers that responded, we had 37 that were certified organic farms. About 85% were enrolled in milk recording. I think the national average is somewhere between 75 and 78%, Danielle. So I think that's maybe a little bit overrepresented in milk recording herds. About 60% tie stall, 40% freestall, and we had some bedded pack and other forms of housing as well. And about 12% of farms were using robotic milking systems. So again, a good representation across the industry. Based on the responses, and one of the questions we asked producers in the survey was, would you be willing then to participate in a follow-up exercise, and that be entertain a farm visit during the summer of 2015? Over 80% of the producers said that they would be willing to do so. We organized those regionally to make sure, again, we had farms in all 10 provinces. We identified and trained teams of students, and they really did three things. The first thing they did during these farm visits was to validate some of the questions that had been asked. So to see whether producers were actually doing what they told us they were, and by and large the news there is good. To implement some aspects of the pro-action animal care assessment, and we'll talk about that a little bit more detail later on. So again, to generate some benchmark data in that context, and finally to collect some biological samples for some disease testing that we were going to do, that we are in the process of doing. So to share with you some of the results, and again, these are early on, but one of the areas of focus and one of the areas of interest was biosecurity. Jocelyn Dubuc and his team at the University of Montreal are sort of spearheading this, and Sheikh Rahman is a graduate student, a PhD student with them, quite interested in looking at the implementation of biosecurity practices. I've already told you that about 47% of farms had brought at least one animal onto their farm in 2014. 27% had had animals that had left the farm and returned to the farm for things like shows and those types of things. Diseases that farmers had on farm and were interested in implementing control programs, Staph aureus, digital dermatitis, and third on that list was ringworm, which is interesting. Diseases that they didn't have and were actively trying to prevent from arriving at their farm. BVD was at the top of the list, Johne's disease was there, and then third were foreign animal diseases. Foot and mouth disease was commonly mentioned. We asked, and about 45% of the farms had participated in a voluntary Johne's disease program in one of the programs regionally. So again, about where we'd expect from a national perspective. The other thing that we wanted to look at, and I indicated that during the farm visits, we implemented part of the pro-action animal care assessment. Because of the interest in animal care and animal welfare, we decided, well, if we're going to measure this, let's use the same protocols, the same processes that ProAction was going to use, so that we could then perhaps establish some benchmarks and figure out where we're going to go as a dairy industry. From the perspective of lameness, the bar on the left is for free stalls, and there we were measuring limp or no limp. Recognize that by limp we meant even very slight and subtle limps, so we used a very low threshold for identifying cows with a limp. On the right-hand side is the bar for tie stalls, and there we use the 4 behavioral indicators that, again, are part of the ProAction assessment, and to be positive, an animal had to display 2 or more of those. So overall, we see about just under 25% on average of cows with a limp in free stalls, and just under 35% in tie stalls. Virtually identical to what some of the other reports have been and what they saw in the Dairy Cluster 1 project. And so, again, I think that's a reasonable representation of where our industry is currently. I think it's important that we benchmark this, and I think it's important it gives us an opportunity to show that we can move forward and that we've got an opportunity for continuous improvement. Just to wrap it off, I'll ask a second question. So the 371 farms that we visited during the summer of 2015, one of the other thing that we were scoring were hocks, and so whether the hocks had injuries or not, and by injuries we meant anything that might be a scab or an open wound and moderate to large swelling. And I'd ask you to indicate to me what you thought the response might be as far as how many of those farms actually out of the 371 didn't have any abnormal hocks. Was it none? Was it less than 10%? Was it 10 to 50%? Or sorry, 10 to 50 farms, or 50 and 100 farms? So this is actually the number of farms out of the 371. Okay, it seems to be settling in. Looks like the most common response is B, less than 10 farms. Well, in fact, the news is good. It's between 10 and 50. It's actually 43 farms of the 371, so about 12% didn't have a single unacceptable hock. I think that's excellent news. Again, just a representation of the data, recognizing that the ProAction target is 10%. Our median value there was just under 20%. On the one side there, we have those 43 farms without any hock injuries, and we see it's a general progression. Again, from a benchmarking perspective, I think that gives us an opportunity to understand where we are, and through continuous improvement, move our industry forward, and I think that's exactly what we want to do through ProAction. I think the fact that we've got farms that don't have any hock injuries, I think we need to understand why that is, and actually go to those farms and use those farms and those as good examples to move our industry forward. I'll wrap up by saying thank you. Again, I had quite a nice slide of all the collaborators. I mentioned some of them already. Kathy Bauman was a postdoc who actually orchestrated the whole thing. We had a number of graduate students and undergraduates that were involved in the project, and certainly want to thank Dairy Farmers of Canada through the research cluster for their support, and also for the milk boards for the opportunity to do this. Thank you very much.
Well, I was going to say our next speaker doesn't need any introduction, but I will introduce him anyway. Dr. Herman Barkema is an NSERC Industrial Research and Infectious Diseases Chair. He is also a guest professor at Ghent University in Belgium, and Dr. Barkema's research program focuses on prevention and control of diseases in birds. He has published more than 225 scientific manuscripts and book chapters, and has lectured all over the world. And so I welcome Dr. Barkema.
Thank you, Shelley. It's a pleasure to be here, and I just mention one person, Rejan. It's great seeing you here. So today I'll talk about infectious diseases, antimicrobials, biosecurity, and what the link also is with human connection, a little bit about the latter. So first, infectious diseases. what is the situation that we have in Canada? How do we prioritize diseases to monitor? What is the antimicrobial, the antibiotic situation, and how can we manage with less antibiotics? So if you nationally look, we have quite some trade of animals and animal products. Dave just showed you that 50% of the farms about in a given year buys animals. If you look in a 5-year period, it would be closer to 90%. So there's a lot of animals going from farm to farm. And we have new diseases emerging. I really hope that we won't get Smallenberg virus from Europe. Think of, it seems like long ago, but Mortellaro disease, which caused hairy heel warts, hit us about 30 years ago. So we're getting new diseases all the time, and we have not eradicated even one of them in Canada. And we don't have programs for disease to eradicate. Yes, we have a leucosis program with currently, I think, 23 farms who participate in Canada. So one exception, of course, is Johne's disease. Dave showed 43% of the farms have participated in a Johne's disease program in Canada. So always realize diseases are bought and paid for. Yeah? They go from farm to farm. The best introduction, the best way to get a disease is buying a cow. But also cattle shows are a source of infection. So we should look at what diseases we want to get rid of. But then we also need to change our practices, because freedom of disease is a mixed blessing. If you are free of a disease, the odds that you will get it is of course high if you do cattle trading and other practices that, that make you vulnerable for introduction of diseases. Fortunately, we are getting traceability. We are so lucky that foot and mouth disease has not hit us in North America. Because without traceability, we cannot control an outbreak of a foreign animal disease like that. So, and that's an important part of proaction. So I really, really like proaction. And I put an underline in the word demonstrate. Dairy farmers are doing a good job, but they need to demonstrate it. This is something that, there will be a proportion of dairy farmers that will see it as another bureaucratic measure, but it is so good that the boards of the provincial organizations and Dairy Farmers of Canada agree that this is a priority, building on Canadian quality milk. So if you look at the herd level and biosecurity, there's quite a different approach. When I came to Canada, I was used to eradication programs, not controlling everything with vaccination, like is done in North America. And we all know that vaccines are not 100% effective, and some of the vaccines are just a little bit better than water. So what is most important if you control diseases is management. It's the mindset of getting rid of it. And then, yes, a vaccine can help you getting rid of a disease. And then closed farming is an important part of that. And then, or just say, don't forget the bulls. That one cleanup bull that is bought in a year. That one also can bring diseases in. And then, of course, we have to limit it, the within spread of pathogens, like contagious other pathogens, like Staph aureus within a farm. So a lot of diseases are also spread from cow to calf, like BVD, leucosis, Johne's disease. So realize that when you have diseases on your farm. We just finished a trial where we looked also at calf-to-calf transmission of Johne's disease. And it proves to be very important. We put calves with each other, we infected some, and put calves that were negative with those calves. 7 out of the 14 calves that were not infected picked up the infection from the other calves that were shedding. So calf-to-calf transmission can be very important. So the study that's led by Dave is so important, because if you don't know where you are, you can never decide on where you should go to. And we have in ProAction certain targets that we need to, on-farm levels that we need to pick. Now you see that with lameness, that actually the percentage is higher than the target that we have. So there's work to do. Also because we have farms with lower prevalences. We have farms, like Dave said, without hog lesions. So we need to learn from those farms and implement the measures that they practice. There's a reason why they don't have it. So then you can prioritize if you know what you have. So Canada is absolutely not a front runner in the eradication of disease. We're a lot better than the US. The US is, to be honest, an underdeveloped country from a dairy perspective, in my view, if you look at disease control. But we are doing better, but not as good as some countries in Northern Europe. Leukosis, Northern Europe is free from leukosis. Actually got it in '85 from Canada with semen. And then eradicated it again. Leptohardjo, the same, leptospirosis. Leptohardjo causes milker's fever, a human disease. So if you have milkers or you yourself get very tired and you get muscle pain or neck pain, that could be because of leptohardjo. We have quite some people in the dairy industry that became incapacitated because of leptospirosis. And we don't have a clue how many farms actually are infected. Salmonellosis, a reportable disease in a lot of the provinces. Neospora caninum causes abortions. Johne's disease, and I put an asterisk if it is a proven zoonotic disease. And if there's a question mark, then it's not completely sure. So there are disease control programs in Northern Europe that we can use. So and there are a lot of concerns in the human population about diseases coming from animals. We get disease after disease that comes from animals, not from dairy cows, particularly. BSE was the last one, and that was not important in Canada, but look what happened in the UK. So we have not eradicated one, and we only get new ones. And we have biosecurity programs, but we need to make people understand why we do certain measures. Prevention of foot and mouth disease is something completely different from prevention of digital dermatitis or of Staph aureus mastitis. So biosecurity is a nice word, but we need to look at specific measures for specific diseases. And it's often used by people that don't have a clue of what it really means. So it needs to be disease specific. And we need to prioritize the diseases to control. I think a couple of broad strokes: zoonotic potential, transmissible from animals to humans, is a big one. We should really target those. Also to protect ourselves in the dairy industry. Then large economic impact, of course. And then comes the possibility to control. And we picked one of the most difficult ones, Johne's disease, to start with. But we did that because of the possible zoonotic potential and the large economic impact. And we better pick ones that we or can eradicate and where the prevalence is really low, or the prevalence is really high and it causes a lot of damage. The diagnostics better be good, and it's good if we have measures that are easy to implement. And then comes genetic selection, and to be honest, we don't know a lot about genetic selection of diseases in dairy cattle. There's a lot to be learned there. So the programs exist, and they typically consist of management practices to prevent infection. You need a large number of farms participating to control a disease. It doesn't make sense to have 23 farms in a leucosis program, because if they buy cows from other farms or go to shows with other farms, then the risk that they will get infected again is pretty high. We just need to start making national programs based on data like we are collecting in the National Dairy Study. Good, I'll switch to the other topic, antimicrobial resistance. There is an enormous pressure from human medicine to reduce the use of microbials, antibiotics in livestock. I'm professor in the Faculty of Medicine as well, and this is the one question that I get asked often. What do you guys do to prevent antimicrobial resistance? If you look at the Ontario Medical Association statement on prevention of antimicrobial resistance, 6 out of the 10 statements were about use in animals. If you look at our industry, the majority of antibiotics used is on mastitis, and the biggest one is blanket dry cow treatment. So to reduce antibiotics, most North European countries now switch to selective dry cow treatment. Of course you will hear, yeah, you know, we are not the only one doing it. Yeah, it's also the humans, and resistance to penicillin was present since the discovery, and it's not increasing. And misuse in humans is rampant. How many people get antibiotics for a flu? While, you know, it doesn't work against viruses. But that doesn't matter. Yeah? It is the perception. And they're going to win in the human medicine anyway. So we better get our act together and think about reduction of antimicrobials, like other countries are doing. Because we want to maintain antibiotics for use on animals that are diseased when we really, really need it to keep our social license. So we should develop a target reduction, and I'm a mastitis guy. I always advocated for blanket dry cow treatment, but it won't be possible in the next decades. So we need to reconsider it. Like they did in the Netherlands and other countries. In the Netherlands they just started in 2010 with a reduction, and they achieved a reduction with 70% in the last 5 years. So it is possible. And that was possible because of a collaboration of different organizations, including the dairy industry. But the incidence of clinical mastitis went up a little bit. So there is a trade-off when you do those kind of things. Which animals should be dried off with antibiotics and which ones not? So what should we do? We have organizations, the majority of you is with Valacta or Canvas DHI, and they have shown that they can offer diagnostics to monitor diseases. Like determining which cows are infected or have a high somatic cell count before drying off, so that we can do selective dry cow treatment. We need to do those prevalence studies like we are doing in the National Dairy Study. And frankly, we need to repeat them all the time to see what the progress is. Yeah? Are we progressing, yes or no? So, and you need to determine what your priority is with the diseases and pick them. But pick ones that will have a high participation and then make it happen. Only monitoring is insufficient. And you need to have also advisors that are up to date on how to control the disease. So the possibilities are endless. We're well behind Northern Europe. Like I said, ProAction is a great start, but it is a start. We need coordinated programs on disease prevention and control. We need to be thinking about reduction of antibiotics, and we need research. And it's always dangerous when a researcher says that we need more research, but we need to know when we are doing selective dry cow treatments, what farms and what cows we should use it and what cows we don't have to use it on. And we don't right now. And we need to work together on this. So I just want to acknowledge all the funders of my research chair. It is so good how we work together in Canada to fund research. When we come here as researchers, we feel like coming home. This is where we belong, and we work together on this. Oh yeah, we stimulate you every now and then, and you stimulate us also. But this is such a good place to work, so thank you for that.
Our last presenter, Simon Dufour, Université de Montréal.
Simon Dufour from the University of Montreal. Simon Dufour teaches epidemiology at the Veterinary Medicine Faculty at the University of Montreal. His research deals mainly with mastitis. He's also director of the Canadian Research Network on Bovine Mastitis and the Quality of Milk, a network of researchers made up of 35 researchers, scientists, with 9 universities and a research center of Agriculture and Agri-Food Canada. Thank you. Thank you for the invitation to make a presentation to you. I would mention that when we received the invitation, we were delighted. Dave, however, did say, why did you put me on a panel with such a dubious panel, a dubious group of researchers? So who are the dubious individuals on this panel, I wonder? So my presentation is about mastitis. The title of the presentation, Mastitis, a Complex Problem, Multiple Solutions. Why a complex problem? Well, first of all, when we talk about mastitis, we're talking about 2 health issues. Subclinical mastitis, inflammation, but without any visible signs, and most often it's measured with somatic cell counting, but we also talk about clinical mastitis, so inflammation with visible, outward visible signs. And these are relatively different diseases, and what I'm showing here with this picture illustrates the situation quite well. On the horizontal axis, you see the somatic cell count of dairy farms. Each point represents 1 farm, and on the vertical axis, you see the number of cases of clinical mastitis occurring on these farms over a year. And I would invite you to answer this question. Do you see the same thing as me when I look at this graph? When the somatic cell count increases, do you see more mastitis, less mastitis, or is the relationship between the two not quite clear-cut? Not as clear-cut as that. So here we go. So raise your hand. We didn't include the technology in our presentation. Raise your hand if you think mastitis increases. Raise your hand if you think it decreases. those who find that it's not a clear-cut relationship, well, you saw exactly what you had to see with this graph. There is no clear relationship between clinical mastitis and somatic cell counts. So you see that some farms have very low somatic cell counts, but a lot of clinical mastitis cases, and a lot of farms it's the reverse. And there are farms where, unfortunately, the two problems are prevalent. So these are very different health problems. The other problem is that, in fact, there are 20 or 30 different bacteria that can cause mastitis. So not only do we have two different diseases, but we can make 20 or 30 ways that. So when we're talking about mastitis, we're really talking about 50 or 60 syndromes. If you take leukosis, it's one virus. Iones, it's one type of bacteria, but here there are all sorts of different bacteria that intervene, and they all behave differently. So coming back to the title of this session, Shelley said, "Are we winning the battles but losing the war?" That was the title, and I can tell you that for mastitis, given the complexity of the problem, we'll never be able to win the war and completely eradicate this disease. If upon leaving here, the board of DFC says, "Listen, we're going to give you $5 million for your research in the next few years, and you'll put an end to mastitis," I'd go to bed very stressed out because I know I won't be able to do this. It's not a matter of money. We can win a lot of battles. We're finding all sorts of solutions, and there are battles to be won, but not the war. So here's the research that we've been doing in our network. In the dairy cluster, there were 11 projects that were funded. 9 different universities were involved, researchers from all over the country. We work very closely together in these projects. And there's also participation on the part of agriculture and agri-food, and we have international partners from Bristol, Ghent, and Utrecht. I couldn't, in the span of 12 minutes, talk to you about all of the 11 projects, but I'll talk to you about the results of 2 projects more in depth, 2 very different projects to give you an idea of the breadth of our work. So a more laboratory-based project, more fundamental basic research, and another project more applied at the farm level. And then I'll flag the other projects that we're working on just to give you an idea of what's going on within the network. So the first project I'm going to explain is for the development of a new treatment. It's an antibiotic against Staphylococcus aureus. It was initiated by François Malouin from Sherbrooke University with his colleagues, and it's a project that in fact began in 2006. And it was very fundamental research initially. François wanted to determine what virulence factors intervene, and I'll explain to you what I mean by this. What virulence factors are used by staph in other infections? Well, virulence factors are the tools that the bacteria must use to provoke the infection. And the primary objective of this research was this. If we were able to identify which tools are necessary, we might be able to develop a vaccine targeting precisely those factors. That was the idea. And this project evolved very well over time. And I can tell you that in January, less than a month ago, an agreement was signed with a pharmaceutical company. It was supposed to be made official by today, but it's not yet official, so I can't give you the name of the company. But the technology has been adopted, and they're going to try to develop a vaccine that will be commercially available soon. But in fact, that's not what I'm talking about here. I'm talking about a new antibiotic, because during this research, initiated in 2006, there was a haphazard discovery that was made. They discovered a new class of antibiotics. When we talk about a new class, we're talking about an antibiotic with a new mode of action. To give you an idea of the importance that has, I would ask you to answer this question. Since the 60s, how many new antibiotics have been developed? 2, 5, 10, or more than 20? So 2 is the most frequent answer, but some people think that 5 to 10 might be a good possibility. But indeed, only 2 new classes of antibiotics have been discovered since the 60s. So in the first half of the 20th century, there was an awful lot of development, all sorts of classes of antibiotics that were discovered. And over the last 60 years, virtually nothing new has been discovered. So all the new antibiotics you see emerging on your farms are members of this class. They're changed, they're slightly modified, but they target the same classes of bacteria. And these two antibiotics, in a scientific magazine published in Quebec, were highlighted as really being important developments. There's very little resistance with the treatment that has been established, and we tested it with the first mice and then cows. With regard to the dairy cluster 2, we wanted to improve the molecule, make it more stable but more effective, and then we wanted to confirm the efficiency and safety of the new molecules and their safety for dairy cows in particular. In both cases, we saw that there were molecules that were effective. One of them was more effective for dairy cows, and it was also very safe. So the project is ongoing. There's an agreement with Elanco, a pharmaceutical company, and Elanco with NSERC is funding this project in order to be able to market this product. 2 students have been trained. Veronique Bailey, you see her to the right, and Amarasi, who isn't pictured here, and François Malouin in the middle. So this is a very basic project, but you see the potential. This began in 2006, so it took virtually 10 years to achieve something, to have products that might be marketable. This also illustrates the importance of investing in basic research, because it can lead to really worthwhile, interesting applications and new products. The second project is more applied, much more applied. It's a study of clinical mastitis and repeat cases. It's a project that I am responsible for, and it began just a year ago. So I'm just presenting preliminary results. This is based on the historical data that we gathered with the cohort of dairy farms that we followed for 2 years in 2007-2008, about 100 dairy farms in Canada. And those who are aware of this study might remember that we looked at all the bacteria that had been collected, and we're continuing to work on this bacteria and on the database. So dairy farmers know that when a cow has a clinical case during a lactation, virtually 1 out of 3 will have a second, a third, or a fourth case during the same lactation. It's very frustrating. It's always the same cow. It seems she never develops enough immunity. So we wonder what the problem is. Is it the cow that has a problem? She's too susceptible? Is it the pathogen, the bacteria, that is too resistant and that survives in the mammary gland? Or is it the management of these infections that's in question? So the results I'm giving you today outline what we saw at the level of the cow herself. So I'm giving you data here. On the x-axis, you see the number of days after the first case of clinical mastitis. So right after that case, no cow had an immediate repeat, and then we follow them over time and we see repeat occurrences. And more cows have second, third cases of clinical mastitis in sequence. What you can see is that when it's a first lactation, the repeat incidence is much lower. First lactations have less incidence of mastitis and therefore less repeat cases. We also saw that days in milk are very important at first occurrence. If the first occurrence happens during the first 2 weeks, generally there's less of a risk of a repeat, a relapse. However, repeated cases occur more quickly. If a cow has mastitis in the first 2 weeks of a lactation, the risk of a repeat quickly is high, but they will have fewer cases throughout their lifetime. This indicates that clinical mastitis in the first 2 weeks of a lactation will determine a very different path for the cow throughout that lactation and following lactations than if the first occurrence occurs later. A student is working on this, our smiling Hossein Jamali. You see that our students are very happy individuals. In the next years, we'll be working on the bacteria, on management as well, farm management, so there'll be further results. So, a quick overview of the other projects without presenting the results. We have a project led by Pierre Lacasse, and you see Samuel Longteau, the student in the center. They want to develop a treatment to attract somatic cells at dry-off to the very tip of the udder teats. So the idea is to develop a treatment that could also be used in parallel with conventional antibiotic treatments. We have another project led by Dr. Hua, who is here, and his student. And the aim here is to develop new treatments to target the quarters of the udder that are really infected. We hope to reduce by 50% the use of antibiotics. We said that antibiotic use is very important and we want to reduce this. So this is one such project. We also have a project on new diagnostic tests to be able to predict if a Staph aureus is more likely to remain and become chronic, or if we could treat it, because there's variability with Staph aureus. So we want a better diagnostic test. We have another diagnostic project to validate a technology that presently exists in human labs and that might help us identify mastitis bacteria quicker, less expensively, and more precisely. Another project with Trevor DeVries. I know he's somewhere in the room. I saw him earlier. Here we're dealing with stalls, the design of the stalls, and the bedding used to determine what is best for the comfort of the cows and for udder health. And Coralie presented her project in her video clip, and it deals with molecules that could disperse the biofilm that our bacteria in the mammary gland produce. I'm close to the end. Finally, Herman with Diego, his student, to identify management practices that might have an impact on antibiotic resistance by staph. A project on the cost of mastitis in Canada. We have estimates that we give you regularly, $400 million. They're 10 or 12 years old, so it's time to update this information. We'd like to develop an economic model to explain what the costs are and see what the benefits, impacts are, the cost-benefit of certain management practices. And finally, the last project that we're working on with Dr. Kilton, it's to identify the obstacles to the adoption of best practices, because there are a lot of best practices that could be applied that aren't, and as Dave was explaining, some practices aren't popular at all and aren't being very much used, especially in the area of biosecurity. So those are the projects I wanted to outline for you, and I wanted to spend a few minutes on my final slide to mention a few things, a few accomplishments that we're really proud of. In fact, as Urban was saying, and I support him in this truly, we're always very honored by the confidence you place in us as dairy farmers. You give us these monies, you invest in research. It's always an honor for us, and we try to be as rigorous as possible in the administration of these funds, to do the best research possible and to try to do more with what you give us. Over the last few years, we looked at what was coming down the pipe. We were successful with certain programs. Last year, we were able to get a grant of $1.6 million. 80% of this money will be devoted to research grants in dairy production. We're very proud. Dave and Herman, too, were part of that application. We're really proud because our contribution through research is good, but also to be able to contribute by seeking other monies outside. Well, we're giving back to the industry in this way. It's a way for us to give back, and we're very proud of this. David Weens mentioned this morning that investment is $3 per dollar invested. And thanks to this, we brought it to a 1-to-6 ratio. And we're really very proud of this accomplishment. We have another project that was dear to our hearts. We led this project with Dr. Roy for the development of a mobile application in order to apply a lot of the knowledge generated over the last few years to facilitate its application on farm. It is a $750,000 project with several partners, Velacta, MVPQ, PLQ, Zoetis as well. And as you know, at the network, the research network, we always try to communicate information, to transfer knowledge so that it's more applicable. And this is a wonderful example, and we're very happy with this project. With that, I wish to thank all of the people who contributed to these research projects, and I'd be pleased to answer your questions with my colleagues here at the table.